A very good morning to all of you. Myself, Dr. Abhimanyu Kumar Jha, and I've come from Institute of Applied Medicines and Research, Ghaziabad. I'm working there as Dean, Faculty of Life Sciences, and Head of Department in Department of Biotechnology at Faculty of Life Sciences, Institute of Applied Medicines and Research, that is IAMR in short, at Ghaziabad, India. Now, I would like to thank Mr. Reddy for giving a fine introduction about myself, and I would like to tell a few points about the previous conference. The previous World Congress on Biotechnology and Biological Studies was held in Delhi, as he told you about that, and that was, very successful in a sense that, we had speakers from, all parts of the world in that conference. In this conference also, we are having speakers from most of the parts, rather all parts of the world. And this year, he has tried to improve upon the, quality as well as quantity of the presentations, because he has gone for very good speakers. So we are having good speakers in this conference too. And I'm sure that everyone is going to have his, technical appetite completely filled after the conference, because the speakers, the kind of speakers which we have, the speakers which I met today morning, they have really the proven leaders in their field, and we can move forward with this gesture. And I can tell you one thing, that these conferences are necessary. Why these conferences are necessary is because science can progress only if the interaction is there. Science can progress only if we are able to interact with each other and we are able to disseminate our research ideas. If we are not able to disseminate research ideas, then in that case the progression of science will be stopped. So for biotechnology to progress as a whole and biological studies to progress in a particular way, we need to discuss our ideas, we need to interact with each other, we need to disseminate those ideas, and finally we need to execute upon those ideas. So with this basic theme in mind, this conference had been started last year, and this is the second World Congress in that series, and I'm sure that we all are going to benefit a lot from this. We all are going to learn a lot from this. Thank you. Now coming to the keynote lecture, as I already told you about my introduction, I'm going to talk about promoter hypermethylation of tumor suppressor genes in cervical cancer in North Indian population. Basically, this is a kind of study which is valid for all kinds of population. Only the players changes, only the genes which are involved in various different populations, they change, because these changes, as I will be telling you, the epigenetic changes we call them, they vary from population to population, because these depend upon our dietary factors which we are eating, as well as the environment in which we are present. And this environment is varying from one place to another. The dietary factors are varying from one place to another. So we can move forward now. Coming to the introduction part, we all know that cancer is a case of uncontrolled cell proliferation, and this is a case in which the cells start dividing without any control. Now, coming to various types of cancers, we know the cancers are of various kinds, like prostate cancer, oral cancer, liver cancer, lung cancer, as well as cervical cancer. There are many more like that, but we have focused our study on cervical cancer, which is a very prominent model. But before that, I would like to tell you a few things about cervical cancer. It is the third most common cancer in women and seventh overall, with an estimated 5,29,000 deaths which were reported in year 2008. Why I have focused upon 2008 is because there was an estimated projection which was expected from this year till 2025, according to the data of GloboCan. This picture has, from then onwards, this picture has turned, worst only, because the number of cases of cervical cancer is increasing year by year, day by day. It caused 2,74,000 deaths in 2008, about 88% of which occur in developing countries. Cervical cancer, it is the most frequent cancer among women in India, and even in developed countries, it is the second most common cancer among women after breast cancer. The early stages of cervical cancer may be completely without any symptoms. That's why it is not very easy to diagnose it initially. And the symptoms of advanced cervical cancer include loss of appetite, weight loss, fatigue, pelvic pain, back pain, leg pain, single swollen leg, as well as heavy bleeding from the vagina. Now, coming to the cancer incidence and mortality, here you can see that the cervix uteri cancer is contributing nearly 5.9% to all the deaths which are happening across the world according to this data. And when we talk about the incidences, it is about 5.9%, and when we talk about the mortality, it is around, you can see this one, 9.2%. So 9.2% of the mortality because of all the cancers is caused only because of cervical cancer. Now, coming to the age-specific incidence rates of cervical cancer in India, you can see the age incidence specific rates of cervical cancer is more. Just a minute, I'll go back. Yeah, you can see this. The age-specific incidence is more in case of India in all the age groups. Now, this green one, as you can see, this is for Southern Asia, and the blue line, this is depicting the picture for the world. So you can see the age incidence rates of cervical cancer is more than the world, and as well as the Southern Asia. Both of them are less. The figures for Southern Asia as well as for the world is less as compared to that for India. And here you can see the estimated number of new cases of cervical cancer in India by age group. In 2008, it was this one, and it is projected to be high by 45% in the 0 to 64 years age group, and it is sure to rise by 86% in the 65 plus age group by the year 2025. Now, as we all know, cervical atrial neoplasia, it is a term used to describe abnormal changes in cervical cells, and it is a layer which is present here. This is uterine cervix just behind the vagina, and CIN may progress to squamous intraepithelial lesion, which is called as SIL, S-I-L. It is a condition that precedes cervical cancer, or to carcinoma in situ, and high-grade cell and carcinoma in situ may progress to invasive carcinoma. But the problem is that most of this cervical cancer consists of squamous cell carcinoma, that is 80 to 90%. And 10 to 15% of this cervical cancer consists of adenocarcinoma, as you can see there. Now, coming to the stages of cervical cancer, this is a model of cancer which progresses from stage to stage and from grade to grade. You can see there are 4 stages: stage 1, stage 2, stage 3, and stage 4. And there are different grades: 1A, 1B, 2A, 2B, 3A, 3B, and 4A, 4B. So here you can see that when we talk about this, in stage 1, the carcinoma is strictly confined to the cervix. There are 1A and 1B grades. Then in stage 2, cervical cancer extends beyond the uterus, but not to pelvic wall or lower third of vagina. But in stage 3, the tumor extends to pelvic wall or lower third of the vagina and causes hydronephrosis or non-functioning of kidney. Now, when we talk about the grades 3rd A, in the grade 3rd A, the tumor involves lower third of vagina, but there is no extension to pelvic wall. But in grade 3rd B, extension to the pelvic wall and or hydronephrosis takes place. Stage 4 is the most dangerous one. The tumor is extended beyond the true pelvis and has involved biopsy-proven mucosa of the bladder or rectum. And 4A, in stage 4A I'm talking about, the spread of the growth to adjacent organs takes place, and in 4B, the spread to distant organs takes place. That is called as metastasis, in which the cancer has spread to all parts of the body. And at that stage, if I talk about the 4B stage, the survival rate is less than 5%. That's why it is very easy, it's very important for us. It's very imperative for all of us to detect some diagnostic biomarkers which can help us in detecting cervical cancer at early stages. Now, the risk factors. The first and foremost risk factor, which is very common in all kinds of cervical cancer, is HPV infection. HPV stands for human papilloma virus. This is the structure of HPV 16, which is the most commonly occurring subtype in cervical neoplasia, which is followed by HPV 18. Now, the risk factors, as I can tell you, sexual activity at early age, then multiple sexual partners, poor hygiene, uncircumcised male partners, and immunocompromised defense increase risk for genital HPV infection in women. Now, the cofactors of cervical cancer, as I told you, there are certain cofactors, like one of them is smoking. Not only active smoking, passive smoking is equally dangerous. The women who are married to those persons who are actively smoking, they are at very high risk of cervical cancer. Then use of oral contraceptives, if it is for more than 10 years continuously, it has been found to be a risk factor. Then low socioeconomic status, early age at first sexual intercourse, and AFP, that is age at first pregnancy, has also been considered to be an important risk factor. And then cervical cancer risk depends upon one more factor, that is sexual history of the male partner, as well as deficient diet. This is also a risk factor, which is very, very important. And the risk of developing cervical cancer increases with age. So this is also an important aspect. Coming to the molecular pathology of cancer. Cancer is caused by alterations in oncogenes, tumor suppressor genes, and microRNA genes. Two sets of genes are involved, as you can see. Proto-oncogenes are present in normal individuals. They are converted to oncogenes when cancer happens. And there is one set of gene, tumor suppressor gene. These are basically active genes in normal individuals. They suppress the formation of tumor, but during cancer, these tumor suppressor genes become inactive. That's why cancer happens. So these are two sets of genes which are regulating whether carcinogenesis will take place or not. Now, there is one more important aspect I would like to tell you about that. That is apoptosis. We all know that it is called as programmed cell death. And this is highly regulated process that allows a cell to self-degrade so that body can eliminate unwanted or dysfunctional cells. There are two pathways for apoptosis. You can see there is one called as intrinsic apoptotic pathway. The other one is called as extrinsic apoptotic pathway. But here our focus is not only on apoptosis. It is mainly on epigenetics. So I'll tell you a little bit about epigenetics. Epigenetic changes are those changes in which there is no change in gene sequence, but the expression of the gene is changed. Now, when we talk about the expression of the genes, if the expression is changed, the formation of the functional protein which is formed, that also changes. Now you can see this. These changes include histone modifications as well as DNA methylation. So you can see that when we talk about histone modifications, we include changes like acetylation, phosphorylation, ubiquitination, and methylation, as well as DNA methylation. The most prominent among all the epigenetic changes is DNA methylation. It is called as the black box of epigenetics. Now, why do we call it as black box of epigenetics? Is because you can see this in normal cells. See, this is the promoter region. When we talk about the promoter region, we have CpG islands in the promoter regions. CpG islands are those islands which are nearly 5 base pairs to even 400 base pairs long, and they are present in the promoter regions. The important characteristic is the GC content is greater than 60% in the CpG island. And here you can see the promoter region in normal cells is unmethylated. It is not blackened, and you can see that it is unmethylated, and as a result the mRNA expression is there. But in cancer cells, this promoter region has become methylated. This is showing methylation. So it has become methylated and the mRNA expression is not there. So this is an important thing. And how this methylation is regulated. There are 3 mammalian DNA methyltransferases, DNMT1, DNMT3A, and DNMT3B. And DNA methylation is catalyzed by specific DNA methyltransferases which use SAM. That is, SAM stands for S-adenosyl L-methionine as the methyl group donor. Here you can see how this process takes place. This is SAM. SAM is regulating the methylation of cytosine. So the methyl group, this is CH3, this one, it attaches at the 5th position of cytosine. You can see this. So this cytosine has now been converted to 5-methyl cytosine. So this is DNMT-mediated methylation of cytosine, and here you can see, this is transcriptionally active chromatin, and after the, action of DNMTs, that is DNA methyltransferases, and HDACs, that is histone deacetylases, you can see this transcriptionally active chromatin turn into a transcriptionally inactive chromatin. Now, the cell cycle regulation. Mammalian cells express two classes of CDKIs. CDKI stands for cyclin-dependent kinase inhibitors that are distinguished by their, by their CDK targets. So there are two kinds of, CDK targets. One is on the basis of CYP family, that is CIP-KIP family of CDKIs. These are universal CDK inhibitors. And the other is INK4, that is inhibitor kinase 4. Cyclin-dependent kinase inhibitors, which are specific for cyclin-dependent kinase 4 or 6 inhibition. So here you can see how it works. P27, P21, and P57, these belong to this family, CIP-KIP family, and P16, INK4A. INK stands for inhibitor kinase. So P16, INK4A, P15, INK4B, as well as there is one more gene, that is P14, that is called as P14 alternate reading frame. So they are, involved as, these regulators, positive regulators, they control this step. So these are several steps which are involved in cell division. You know that G1 is there, then S phase is there, G2 is there, and then M phase, that is mitotic phase, is there. Now, this epigenetic, these epigenetic changes, they can be used for developing a therapy as a cure against cancer. So this can be called as epigenetic therapy. This is the use of drugs to correct epigenetic defects, and this is one of the most recent and rapidly developing area of pharmacology. And these two important enzymes are there: DNA methyltransferases and histone deacetylases. They are the two major drug targets for epigenetic inactivation. And here you can see the demethylating agents. They work like this. They can work like, they can go for covalent trapping of DNMT1. As I told you, DNMT1 is DNA methyltransferase 1 through incorporation into DNA. And there are certain, nucleoside analogs which have been discovered, like 5-azacitidine, 5-aza-2-deoxycytidine, as well as zebularine. And then there is, non-covalent blocking of DNMT1 catalytic active site. It can be done through EGCG. This is found in green tea. So this is very, very important one. And then there are several other mechanisms. So I'm not going into the detail of these mechanisms, but the important point which we should keep in mind is that two nucleoside analogs have been discovered, like 5-azacitidine and decitabine. They have recently been approved by Food and Drug Administration for the treatment of myelodysplastic syndrome. And due to the cytotoxic effects of these, demethylating compounds to normal cells, now the focus has shifted to discovering some oral compounds, some natural compounds, some dietary compounds which we take in our diet to screen them for the potential to cause reversal of hypermethylation of the promoter region of these genes, which can actually lead to a reactivation of these suppressed or downregulated tumor suppressor genes. Now, coming to the international status, these are the things. Aims and objectives. It was to study the promoter hypermethylation of tumor suppressor genes, including P14, P15, P16, P21, P27, P53, as well as we selected several other genes which have not been mentioned here, like FHIT, DAPK, STAT1, RB, that is retinoblastoma, in cervical biopsies and serum samples. And this was done in age and sex matched controls. And to study the correlation of methylation status of the tumor suppressor genes with respect to stage of disease as well as HPV infection. Now, these are the material method. We collected 125 biopsy samples and paired blood samples from Dharamshila. A few samples were collected from Dharamshila, but most of the samples were collected from Mohandai Oswal Cancer Treatment and Research Foundation, Ludhiana, after obtaining the necessary ethical clearance. And biopsy samples were collected. Then, blood samples were also collected from healthy females. They were used as control. And biopsy samples, a few biopsy samples were also used as controls because they were collected from healthy females in which hysterectomy had been carried out, but cervix was normal, and control samples were age matched with the patients. Now you can see this was the methodology which was used. Samples were collected, and then the DNA was extracted from biopsy and blood samples using PCI, that is phenol chloroform extraction. Then one part of the study was focused on HPV infection and HPV16 typing. The other part was focused on the methylation study. So in this, we carried out bisulfite modification, which was followed by MS-PCR using methylation-specific primers. We use methylation-specific primers in this, PCR, it uses two kinds of primers. One is unmethylated primers, the other is methylation specific primers. And the purification of MS-PCR product by gel extraction, using this gel extraction kit, and this was followed by sequencing. When we do MS-PCR, this MS-PCR is a very sensitive technique. We need to understand that it can detect even 1 in 10,000 allele if it is hypermethylated. And there is one more thing. This MS-PCR will detect the change which has been carried out by bisulfite modification. After bisulfite modification, unmethylated cytosine gets converted to uracil, but methylated cytosine is not converted to uracil. This change is detected by this MS-PCR. And coming to the RNA part, because we needed to understand, we needed to see whether this promoter hypermethylation results in downregulation of expression of these genes or not. So we carried out the RNA work also, RNA extracted from biopsy and blood samples using tri-reagent, sigma-L-rich, and complementary DNA, that is cDNA, was prepared using Rivertate first strand cDNA synthesis kit from Fermentas, and semi-quantitative RT-PCR and quantitative real-time PCR was carried out, which told us about the changes in gene expression, the change in how many fold change which was there in the gene regulation or the expression of the genes. Next is this is the basis of MS-PCR, which I have already told you. Now coming to the statistical analysis, we obtained data stratified and classified for better interpretation, and the reported odds ratio could be interpreted as age-adjusted estimates of relative risk of developing cervical cancer, and then the amount of RNA expression was obtained by grading a ratio between densitometry results of test gene and beta-actin. And an important point which is to be noted is p-value less than 0.05 was considered as significant. We calculated the gamma coefficient to correlate the hypermethylation of tumor suppressor genes with increase in stage of cervical cancer. So there were several things which were there. And then coming to the results part, you can see these were the various age groups. We classified our data into less than equal to 45 years and greater than 45 years. You can see this. We classified our data on the basis of the risk factors selected for this study. They were oral contraceptive use, smoking status, and age at first sexual intercourse, as well as the normal age. So this was there. You can see this. HPV infection was confirmed by PCR product of 450 base pair. This is the band for HPV16. You can see this. And then HPV16 typing was confirmed. And this is the PCR product of this. 206 base pairs was there. You can see this. 92.8% patients were found to be HPV positive. As I told you, this is the major cause of cervical cancer. Now, coming to the MSP of INK4A family, you can see that this is the result of methylation-specific PCR of P14 gene. U stands for methylated product, and M stands for methylated product. You can see this. This is the result for P15 gene, and this is the result for P16 INK4A gene. The unmethylated product was 151 base pair. The methylated product was 150 base pair. Here you can see you can have a look at this. In some of the samples, it was found to be unmethylated. Sample number 1 was unmethylated. There is no band in methylated lane. This lane is representing the sample for methylated PCR product, and here you can see sample number, again this one, sample number 5, this is methylated. This is showing the methylation specific band, and even again sample number 6 also, this is showing the methylation specific band. So we calculated how much this hypermethylation of P16 is associated with the increasing risk of cervical cancer, and we were significantly able to show that there is an increasing risk of cervical cancer which increases with increase in hypermethylation of P16 INK4A. Hypermethylation of this P16 gene was observed in 36% of cervical cancer patients, and risk of cervical cancer was increased significantly by 2.19-fold due to this hypermethylation of P16 INK4A in cervical cancer patients. Here you can see this 2.19. So this was a big thing, and now we correlated it with HPV infection, and this was found to be significantly correlating with HPV infection. As you can see that when we talk about HPV infection, p-value was there, which was required, but that's a very statistically significant correlation was not there in HPV infection. And correlation of promoter hypermethylation was age. With age it was done, and it was found to be statistically significant in patients of both age groups, that is age less than equal to 45 years as well as age greater than 45 years. Then now this one, you can see this was for the passive smoking patients. you can see risk of cervical cancer increased significantly by 2.20-fold in association with P69K4A hypermethylation with respect to passive smoking, and the p-value was found to be highly significant. With OC users also, it was found to be significant, and the risk of cervical cancer increased by 2.16-fold in association with P16 hypermethylation in the case of OC users. But in case of P14, we can see that this was not found to be significant. In case of P15 also, it was not found to be significant with respect to oral contraceptive users. Significant association was found in both these age groups, in age at first sexual intercourse less than or equal to 20 years, as well as age at first sexual intercourse greater than 20 years. Now, these are the results of bisulfite sequencing here. You can see this is this C. This is the one which is representing the cytosine, and this is the bisulfite sequencing, which revealed conversion of unmethylated cytosine but not methylated cytosine, which could downregulate the expression. So this is for P14, this is for P15, and this is for P16INK4A. Now the RT-PCR was carried out to see what impact this promoter hypermethylation is having on the change in gene expression. You can see this, the transcription level was significantly reduced in patients with methylated promoter in comparison to patients with unmethylated promoter, but this, in P14, the downregulation was 31%. In P15, the downregulation was found to be 20.2%, but in case of P16, the downregulation was found to be 85.3%. Now, this was a remarkable thing, and the p-value also was found to be highly significant. That was 0.000. Then we went for doing this real-time PCR, and this quantitative real-time PCR results showed that around 6.02. Now, this is the calculated fold change, that is change in delta CT value. You can see after methylation there is 6.02 fold downregulation. It was a mean of 3 of P16INK4A mRNA expression due to hypermethylation of promoter region of this gene. Similar studies were carried out for other classes of families also. Here you can see P14 could not be used as a significant biomarker for diagnosis of cervical cancer because it was not found to be significant with increasing the risk of cervical cancer. P16, it was found to be statistically significant even in case of passive smokers as well as OC users, and this is the first report on significant hypermethylation of P15INK4B gene in cervical cancer. So my group was able to confirm it for the first time that this P15 gene is involved in the hypermethylation of this gene is involved in the progression of cervical cancer. We reported it in 2012, and there was a paper in Molecular Biology Reports about this. Then there is MSP of CIP and KIP family. You can see this P21, CIP1. This is the methylation specific product. Then P27. Again, you can see this is the methylated sample, and this sample was unmethylated. This is P57. Now, the frequency of promoter hypermethylation, none of these genes were found to be statistically significant with respect to HPV infection, age group, passive smoking, use of oral contraceptives, as well as age at first sexual intercourse with respect to increase in the risk of cervical cancer. Now, coming to this one, here you can see the bisulfite sequencing data, same thing, which was carried out for these genes as well. And we calculated the gene expression in case of these genes also. We just wanted to see, although the significant hypermethylation was not there, but we wanted to see how much downregulation in expression was there. So we were able to see this 24.6% downregulation was there in case of those genes, in case of those samples in which hypermethylation was found. And 23.7% downregulation was observed in case of P27, and 20.2% was observed in case of P57. So then we went for carrying out MSP for P53 gene. Now this gene is a very important gene because it was originally discovered as a tumor suppressor gene, and this family of genes include P63, P53 as well as P73 genes. Now when we talk about this gene, you can see that, for example, in sample number 1, this sample was found to be unmethylated because the band is there in the lane in which the samples for unmethylated specific PCR were run. There is no band in lane number 2 in which methylation specific band should have been there if this person would have shown hypermethylation of P53 gene. But here you can see in sample number 5 the methylation was there, in sample number 4 the methylation was there, and this was the study was the first study which showed that P53 hypermethylation is observed in case of cervical cancer in North Indian population. Because we were the first one to report that We reported it, I think 5 to 6 years back. And along with this, we carried out our study on P73 also. You can see this. These are the bands for unmethylation specific PCR as well as for methylation specific PCR. And this is the bisulfite sequencing data of P73 representing the cytosines. C is in blue. Now coming to frequency of promoter hypermethylation, both of these genes showed, significant, hypermethylation, and you can see the p-value was found to be less than 0.05 in both the cases. It was found to be highly significant in case of P53 as well as P73 genes. And when we talk about this one, you can see significant risk in increase of cervical cancer with respect to P53 hypermethylation was observed in OC users, that is oral contraceptive users, whereas significant increase in risk of cervical cancer in association with P73 hypermethylation was observed in case of passive smokers. Coming to the RT-PCR, here you can see after P73 hypermethylation, that is promoter region hypermethylation of P73, you can see the percentage of downregulation was found to be 69.1%. This was the data from semi-quantitative reverse transcription PCR, and this was found to be highly significant with a p-value of 0.000 till 3 decimal places. And here you can see the result of quantitative real-time PCR. The downregulation of P73 mRNA expression was around, you can see this, 4.5-fold due to hypermethylation of promoter region of the gene. Now the crux was, this is the first report with respect to methylation of P53 in cervical cancer patients. Significant trend towards increasing risk of cervical cancer was observed with respect to methylation of P73 gene, and hypermethylation of P73 was found to be significantly increasing the risk of cervical cancer among passive smokers. Now the study which was carried out on RAR beta 2 gene, this is a retinoic acid receptor beta 2 gene, and here you can see significant hypermethylation was observed. This sample number 5 is methylated, 6 is methylated. We carried out studies on so many samples, and the reported risk ratio was 2.29 with 95% confidence interval of 1.92 to 2.74, and the p-value was found to be highly significant, 0.000 in this case. And the bisulfite sequencing also really confirmed our results. This gene is really very important in the progression of cervical cancer. The hypermethylation of this gene is significantly important. So later on, when I carried out my, as I told you about the epigenetic therapy part, when I wanted to carry out the study on epigenetic therapy, when I wanted to see whether these changes can be reversed, because unlike genetic changes, these epigenetic changes can be reversed. So when I planned that study, I selected this gene for that study. And I was able to successfully show that after treatment with natural compounds, the promoter hypermethylation of this gene, RAR beta 2, gets reversed, and that leads finally to reactivation of this tumor suppressor gene. Now here you can see the hypermethylation of RAR beta 2 was observed in 40.8% of the patients, cervical cancer patients, and statistically significant relation was detected with respect to passive smoking as well as oral contraceptive use, that is more than 10 years. And the risk of cervical cancer was increased around 2.33-fold in both the cases. Here you can see 2.33 in both the cases. Now carrying out the RT-PCR, it was a mind-blowing 90.2% downregulation was detected after semi-quantitative reverse transcription PCR, in case of methylated RAR beta 2 promoter, and the downregulation of RAR beta 2 mRNA expression was found to be 11.8-fold. Here you can see that, due to hypermethylation of promoter region of this gene. Now, another gene which was used in this study was fragile histidine triad, FHIT. FHIT stands for fragile histidine triad gene. And here you can see the samples which were methylated. Clear-cut demarcation was there. And, this gene was found to be hypermethylated in nearly 37.6% of the population out of the 125, samples. You can see 47 were found to be hypermethylated, and the odds ratio was 2.22 with 95% confidence interval of 1.87 to 2.64, and the p-value was found to be highly significant. The correlation of promoter hypermethylation of FHIT with risk factors showed great correlation. The risk of cervical cancer was increased around 2.5-fold in case of passive smokers as well as OC users. Now coming to the semi-quantitative reverse transcription PCR, the transcription level was significantly reduced in patients with methylated promoter in comparison to patients with unmethylated promoter. It was found to be 87% downregulation, and the downregulation of FHIT mRNA expression was around 9-fold due to hypermethylation of promoter region of this gene. Then another gene which we selected in our study, which was important, was death-associated protein kinase. Now this is a tumor suppressor gene, but this is helping in apoptosis also. So what we did was, we carried out the same study and found out that this gene was also found to be significantly hypermethylated, and the risk of cervical cancer was found to increase among passive smokers and OC users with the hypermethylation of death-associated protein kinase promoter. Semi-quantitative RT-PCR that showed again high downregulation. It was 88.87% of downregulation after methylation of DAPK promoter, and the downregulation of DAPK mRNA expression was found to be around 6.21-fold due to hypermethylation of promoter region of this gene. Now the crux in a nutshell: significant trend towards increasing risk of cervical cancer was also observed with respect to methylation of RAR beta 2, FHIT, and death-associated protein kinase genes. Hypermethylation pattern of these genes in cervical cancer with respect to HPV infection was insignificant. Why this was insignificant was because HPV infection is the major cause in all the cervical cancer cases. So whether OC use is there or not, whether passive smoking is there or not, whether age at first sexual intercourse is there or not, whether age of first sexual intercourse is less than 20 years or it is greater than 20 years, it doesn't matter. Hypermethylation of RAR beta 2, FHIT, and DAPK, that is death-associated protein kinase, was observed to be significantly increasing the risk of cervical cancer among passive smokers as well as oral contraceptive users. And age and FSI, they were not found to have any impact on the significant correlation of the hypermethylation of any of these genes with the increased risk of cervical cancer. The analysis of semi-quantitative and quantitative RT-PCR showed significant effect of hypermethylated promoter in downregulation of RAR beta 2, FHIT, and death-associated protein kinase gene. And this is the first report on significant hypermethylation of RAR beta 2 and death-associated protein kinase in North Indian population. This is also the first study to show the significant association of hypermethylation of FHIT with passive smoking and OC use, and therefore these genes can be used as significant and reliable biomarkers for diagnosis of cervical cancer. We also carried out our study on RB1, that is retinoblastoma 1 gene, and STAT1 gene, but these genes did not show any significant correlation with the increasing risk of cervical cancer. They were not found to be significantly hypermethylated. So basically, when we come to conclusion, in the present study, 92.8% of the patients were found to be positive for HPV. Hypermethylation of P59K4B showed marginal association with the risk of cervical cancer, but strong association between hypermethylation of these genes, P69K4A, RAR beta 2, FHIT, death-associated protein kinase, with risk of cervical cancer was observed, and hypermethylation of these 4 genes, P69K4A, RAR beta 2, FHIT, and DAPK, were observed to have significant correlation with increase in risk of cervical cancer in association with passive smoking as well as oral contraceptive use, because the p-value was found to be less than 0.001, not only 0.05, it was found to be less than 0.001. And the hypermethylation of these genes were not found to be age-dependent and affected by FSI. Hypermethylation of these genes significantly decreased the mRNA expression, and P53 hypermethylation was found to be significant in cervical cancer patients. P73 was also significant. Then significantly increasing trend for methylation of P73, RAR beta 2, FHIT, and death-associated protein kinase genes was